Welcome back to the Crystal Classroom series. Today we'll be discussing one of the most important aspects of solid-state screening, the factors that label a phase or polymorph as better or worse than another. The first of these factors is solubility. From a Gen Chem perspective, every compound has one solubility in a given solvent. Well, this is true on a geological timescale. On a biologically relevant timescale, it really is not. The solubility of a solid when placed into a liquid is tied to the energy at the surface of the crystal and how it interacts with the solvent. Consider a compound that has a hydrophobic and a hydrophilic region. If it packs in a way that isolates and protects the hydrophilic region, then it's likely creating a more hydrophobic surface, making it less likely that it will dissolve in water. This means that different polymorphs can have different solubilities. While it would be intuitive to say that the greatest water solubility would be the best polymorph, it isn't quite that simple. We'll go into this in more detail another time. But suffice it to say that some degree of water solubility is definitely desirable. Second is what we call the phase origin. This refers to whether a compound is a hydrate, a solvate, or an anhydrate. If something is anhydrous, it has neither water nor an organic solvent within the crystalline matrix. If something is a hydrate, it has a degree of water integrated into the packing structure at the molecular level. If something is a solvate, then it has some organic solvent integrated into its crystalline lattice. Of these three options, anhydrates are the most preferred. Anhydrous phases tend to have higher water solubility than hydrated phases. There is also a question of water sensitivity and the possibility for hydrates to change phases and either gain or lose water if the humidity crosses a particular threshold, called the critical water activity. Anhydrous phases, which do not demonstrate hygroscopicity, which is the tendency to uptake water, do not have a major risk of converting. Hydrates are a viable alternative to anhydrous phases if they have enough stability to avoid phase changes. Lastly are solvates, which may be problematic. A crystalline phase which contains an integrated quantity of a solvent with a known degree of toxicity, such as benzene, chloroform, nitromethane, dichloroethane, or others, already has a steep obstacle towards being considered safe, and with very good reason. Some solvents are more acceptable, since a patient is likely to be able to absorb trace quantities of them without negative effects, such as ethanol, dimethyl sulfoxide, or acetone. Solvents are classified by a set of guidelines for patient exposure by the International Council for Harmonization into three categories. Class 3 solvents are the most benign, while Class 2 solvents have strict limits on a daily exposure and incidental intake. Class 1 solvents are right out. Ensuring the safety and stability of solvates is frequently more trouble than it is worth, especially if there is a safer alternative. Finally, there's a question of stability. This part is often tricky. Stability and solubility tend to have an inverse relationship. The more stable a phase is, the lower its solubility tends to be. However, a less stable phase always has the possibility of converting into a more stable phase if one exists. Long-term stability testing can be a time-consuming endeavor, especially since the stability of an API in early development phases may or may not be the same as a drug product with excipients, so test once or twice. If the most stable phase is selected and the drug product is formulated to try and offset the lowest available water solubility, then the testing might only need to be done once. If a metastable form is selected, then the question about its shelf life and potential to convert to a more stable phase and less soluble form cannot be ignored. The end result is that the ideal active pharmaceutical ingredient is one that has the best water solubility, is anhydrous, and is also the most stable phase. This does have an obvious contradiction in it, which is why compounds with a broad polymorphic landscape make for some very difficult decisions. After all, pursuing development of multiple phases of the same compound costs far more than having one clearly best option.